Hello viewers, I am again here before you with a new topic of genetics, and this topic is transcription. If you like my videos, then please do share and subscribe my channel also. So what is transcription? Transcription is the process of transferring genetic information from one of the strand of DNA, one of the two strands of DNA to RNA. As you know that DNA is a double-stranded structure, and out of the two strands of DNA, there is only one strand which contains information for protein synthesis, and that strand which has information for protein synthesis is called template strand, whereas the complementary strand is called coding strand. And that portion of DNA, that segment of DNA which has information for a protein, that is called a cistron. In the beginning of cistron, there is promoter. In the last of the cistron, there is a terminator. So this segment of DNA between promoter and terminator, this is called a cistron. And this cistron has the information for one protein. This process of transcription, it is also based on the basis of complementarity as the process of replication. But there is one difference. What is that difference? With adenine, there is not thymine. Adenine pairs with uracil. So if adenine is there in DNA, then opposite to adenine, there will not be thymine, but there will be uracil. Now there is question, when DNA is double-stranded, then why only one strand of DNA has genetic information? There are two reasons for this. If both the strands of DNA will participate in the process of transcription, then there will be formation of two RNAs, and these two RNAs will be complementary to each other, and the two RNA which are synthesized from a single DNA, they will wind up and they will form a double-stranded RNA, which will be a useless structure for translation. Second point, if by chance two RNA are synthesized from the two strands of DNA, then they will code for two different proteins. So from one cistron, there will be synthesis of two proteins, but there must be synthesis of one protein from a cistron. That's why from the two strands in DNA, only one strand has information for protein synthesis, and that strand is template strand. The process of transcription, it is divided in three steps: chain initiation, chain elongation, and chain termination. We are studying the process of transcription in prokaryote, in bacteria. There are three types of RNA: ribosomal RNA, transfer RNA, and messenger RNA. For the synthesis of all the three RNA in bacteria, there is a single RNA polymerase, and that RNA polymerase, DNA dependent RNA polymerase, which catalyzes the synthesis of RNA on DNA template. So that enzyme, RNA polymerase, it is responsible for unwinding the two strands of DNA as well as for the synthesis of new RNA on template strand. But for starting of replication, for starting of transcription, for chain initiation, only RNA polymerase is not able. This RNA polymerase is helped with another protein which is sigma factor. So sigma factor becomes attached with RNA polymerase transiently, and these both structure, RNA polymerase and sigma factor, they start the unwinding of two strands of DNA and synthesis of RNA on DNA template. Once RNA synthesis is started, after initiation, the sigma factor is separated, and only RNA polymerase start the process of transcription of RNA. on DNA template. When the process of transcription proceeds and this process reaches up to terminator, then the process of transcription should be stopped. And here, just to terminate the process of transcription, another protein participates here, and that protein is Rho factor. So Rho factor attached with RNA polymerase, and it is responsible for chain termination. So you can see here there is RNA polymerase which is responsible for process of transcription in bacteria. But as far as chain initiation is concerned or chain termination is concerned, RNA polymerase attach transiently with sigma factor for chain initiation and attach with Rho factor for chain termination. In this way, the process of transcription is completed. In bacteria, the process of transcription and process of translation are going on simultaneously, because in bacteria there is no separate nuclear membrane. The nuclear chamber is not totally separated from cytoplasm. Both processes of transcription and translation are taking place in the same compartment. That's why, as the process of transcription is going on, simultaneously the process of translation is also taking place. So that was the concept of transcription in bacteria. Thank you very much.